Hello everyone, welcome to the channel Microbial World. So in the previous session, we discussed about the cells of the immune system. Lymphocytes such as T cells, B cells, natural killer cells, phagocytic cells such as monocytes and macrophages. Today we will be discussing about granulocytic cells and dendritic cells. Granulocytes are classified based on their cellular morphology and cytoplasmic staining characteristics. They are classified into neutrophils, eosinophils, basophils or mast cells. Okay, so as the name says, the cytoplasm of granulocytes is filled with granules, and these granules will be released when they come in contact with a pathogen. Okay. So these granules contain many proteins which has certain functions. These proteins can damage the pathogen directly. And also some of these proteins can regulate the activity of white blood cells such as lymphocytes. And some of them is also involved in remodeling of tissues at the site of infection. Okay. Neutrophils. Neutrophils measure 11 to 14 μm in diameter. It has a multi-lobed nucleus. Multi-lobed nucleus means it is a cell nucleus that has multiple lobes or sections. Okay. So it has a multi-lobed nucleus and a granulated cytoplasm that stains with both acid and basic dyes. It is also called polymorphonuclear leukocyte. That is because of its multi-lobed nucleus. They constitute 50 to 70% of the circulating white blood cells. Neutrophils are produced in the bone marrow through the process of hematopoiesis. Then they are released into the peripheral blood where they circulate for 7 to 10 hours before migrating into the tissues. In response to many types of infections, the bone marrow releases more than usual number of neutrophils. Okay, and these neutrophils are the first cells that arrive at the site of inflammation. And this increase in the number of circulating neutrophils is known as leukocytosis. So, when more number of neutrophils are being released, it indicates that there is an infection, right? The movement of circulating neutrophils into tissues is known as extravasation. So during extravasation, the neutrophils will migrate from the bloodstream into the site of infection or tissue damage. And then the neutrophils, they will adhere to the blood vessel wall. And they will squeeze through the blood vessel wall by a process known as diapedesis. And then they will migrate towards chemotactic signals that is being released from the site of infection. Okay. So once they reach that affected tissue, the neutrophils, they will eliminate the pathogen by phagocytosis or by releasing antimicrobial substances. Okay. So I hope you understood about extravasation. Several substances can be generated during an inflammatory reaction which serves as chemotactic factors. Okay, so during an inflammation, chemotactic factors will be released. Okay, so these chemotactic factors can be peptides, cytokines or bacterial products and all. Okay, and these chemotactic factors will promote the accumulation of neutrophils at the site of inflammation. Chemotactic factors include complement components, blood clotting system components and cytokines secreted by activated T helper cells and macrophages. Neutrophils are active phagocytes like macrophages. Okay, and primary and secondary granules are present in neutrophils, and in this granules, lytic enzymes and bactericidal substances are present. Larger, denser primary granules are a type of lysosome containing peroxidase, lysozyme and various hydrolytic enzymes. Smaller secondary granules contain collagenase, lactoferrin and lysozyme. Both the primary and secondary granules fuse with phagosome and digest and eliminate the contents similar to macrophages. So they are functioning like macrophages. Okay. So this is the structure of neutrophil. You can see it has a multi-lobed nucleus. Then there is primary azurophilic granule, secondary granule. Okay, so what did we say? The primary granule contains peroxidase, lysozyme and other hydrolytic enzymes. Secondary granule contains collagenase, lactoferrin and lysozyme, right? Then there is phagosome and glycogen. Eosinophils. They measure 11 to 15 μm in diameter. It has a bilobed nucleus, which means the nucleus has two lobes, okay, and a granulated cytoplasm that stains with the acid dye eosin red. So you can understand where the name eosinophils came from. It is from this acid dye eosin red. They constitute 1 to 3%. They are motile phagocytic cells that can migrate from the blood into the tissue spaces. Eosinophils play a role in defense against multicellular parasitic organisms, including worms. So, how they are doing this? They also contain granules, eosinophilic granules, okay? And the contents that are secreted from the granules will damage the parasitic membrane, okay? So, this is how they defend against worms. They also secrete cytokines that regulate B and T lymphocytes, thereby influencing adaptive immune response. So what is adaptive immune response? It is a highly specific immune response by our body against pathogens. Okay? It involves recognition of antigen by B and T lymphocytes, where B lymphocytes, they will produce antibodies against the antigen, and T lymphocytes, once they are activated, they will destroy the infected cells. Okay, so this response, it is based on immunological memory, so that our body can act faster and provide an effective response on subsequent exposure. Okay, eosinophils also contribute to asthma and allergy symptoms. Hypereosinophilia that causes organ damage is called hypereosinophilic syndrome. This is the structure of eosinophil. It has a bilobed nucleus and you can see crystalloid granules are also present. Basophils. It has a lobed nucleus and heavily granulated cytoplasm that stains with the basic dye methylene blue. It constitutes less than or equal to 1%. They are non-phagocytic granulocytes. So we said earlier that neutrophils and eosinophils are phagocytic, okay? But basophils are non-phagocytic granulocytes. They contain large granules that are filled with basophilic proteins. So in basophils, many granules are present and these granules contain proteins. They play a major role in certain allergic responses. Histamine is the most common and well-known protein in basophilic granules. They play a role in increasing blood vessel permeability and smooth muscle activity. So like eosinophils, basophils are also important in response to parasites, particularly helminths, worms. Okay? They secrete cytokines that assist in the modulation of the adaptive immune response. They also contain anticoagulant heparin. This is the structure of basophil. It has a lobed nucleus and as we said, it has a heavily granulated cytoplasm. Many granules are present, right? And then there is glycogen. Mast cells. They are formed in the bone marrow. They are released from the bone marrow into the blood as undifferentiated cells. And when they enter the tissues, then they mature. Mast cells are found in wide variety of tissues such as skin, connective tissues of various organs, and mucosal epithelial tissue of the respiratory, genitourinary and digestive tract. Like basophils, they also have large number of cytoplasmic granules that contain histamine and other pharmacologically active substances. They also play an important role in the development of allergies. Dendritic cells. They were identified in 1868 by Paul Langerhans during a close anatomical study of skin. And dendritic cells were the first cells of the immune system discovered. They are covered with long membrane extensions resembling the dendrites of nerve cells. They are antigen presenting cells like macrophages, okay, because they possess MHC class. There are 4 types of dendritic cells known. Langerhans dendritic cells. They are found in the epidermal layers of skin. Then there is interstitial dendritic cells. They are present in the interstitial spaces of all organs except brain. Monocyte derived dendritic cells. They arise from monocytes that have migrated from the bloodstream into tissues. From the tissues, they can move through lymph to lymph node and then cross back into the bloodstream, and then they are transported to lymphoid tissue. Plasmacytoid derived dendritic cells. They arise from plasmacytoid cells. Okay, they play a role in innate immune defense and as antigen presenting cells. Innate immune defense, it is a non-specific rapid response mechanism that will provide immediate protection. Okay, this is the structure of dendritic cells, and as you can see, it is covered with long membrane extensions, right? So this is all about granulocytes and dendritic cells. Thank you.